key challenge of the Madingley model was to explain and simulate how interactions among individual organisms leads to the structure and function of ecosystems at larger scales, like at the scale of the landscape, or even whole continents, biogeographical patterns, that kind of thing. And to do that, we had to make a number of abstractions. How could we define a model at the level of the individual and yet still be able to simulate it at that much higher level? The first choice we made was to not go in with the usual route of Linnaean taxonomy, so to say that species, to look at individuals as different species. So instead we view and treat individuals as individual organisms with measurable functional traits, things like whether you're a herbivore or a carnivore, or what your adult body mass is, so at which body mass are you going to switch to becoming reproductive, what's your juvenile body mass, so how big are the juveniles that you create, as well as your current body mass, whether you're an ectotherm or an endotherm, or whether you're iteroparous or semelparous, which is to do with how you reproduce. But that still leaves us with a real challenge, because in any one place we have a huge number of these individuals. At the scale we typically run the model at, which is something like a 1 by 1 degree grid cell, we have something in the order of 10 to the 14 individual organisms, which is a titanically huge number. So there's no way we could simulate the fate of each of those individuals separately. So the second approximation we use is called the cohort approximation. It's quite a simple idea. The idea is that if you freeze frame that collection of a huge number of individuals, you can then identify packets of organisms which are extremely similar in all of those traits. And then you make an approximation, a leap of faith. So you say, well, now let's aggregate them together and say that they're absolutely identical. And now if you simulate the fate of one of those organisms in that packet, you can then assume that all of the organisms in that packet have the same fate. Now that assumption that they all have the same fate is actually related to another approximation we make, which is called the mean field approximation, which is we assume that all of the individuals within a region are well mixed, so they're all kind of milling around and bumping into each other. And so with all of those approximations in place, we're able to write all of the fundamental rules and equations of the Madingley model at the scale of individual organisms. So like, how quickly are you metabolizing, what do you eat, how quickly are you growing, when do you reproduce, and so on. So at the individual scale, and yet through these approximations we're able to approximately simulate huge numbers of them, like 10 to the 14, and in fact in a particular region, and when we run it for the whole globe, we simulate around 10 to the 16 individuals. Is that all right?